Good evening, everybody. Thanks for joining us this evening. It is my true pleasure to welcome everyone to our 26th season of Sea Secret. I'm Ronnie Avissar. I'm the Dean of the Rosenstiel School. Before I move on, thank you for joining us tonight for this series. And all lectures that we are going to be offering this evening are given by the faculty of the Rosenstiel School to emphasize the 4 strategic initiatives that we have launched a couple of years ago. Those strategic initiatives are based on solving big issues for human and the planet over the next years. So those 4 initiatives we have heard already at our last presentation. One of them is connected to the food, the problem of feeding the world. And we discuss aquaculture activity that is done at the school and around the world. The 3 other initiatives, in addition to feeding the world, include saving lives, protecting our resources, and also unlocking the ocean secrets. This evening, we are going to hear from one of our younger, but certainly not in terms of the science faculty, and I'll introduce her in a couple of minutes. What I would like to do also is give a special thank you for our sponsors of this series. And that includes the presenting sponsor, Bank of America, the Shepard Broad Foundation, Bill Galway, Cheryl Gold, the Key Biscayne Community Foundation, the Key Biscayne Life Enhancement Fund, and the Joan McCann Family Foundation. Thank you all for supporting this series and the school in general. So tonight I have also the pleasure, as it is customary now in our series, to introduce to you Denise Hassinger, who is our Masters of Professional Science program. She is one of the first recipients of the Colonial Lower Memorial Endowed Scholarship, which was created in 2018 in memory of Miss Cornelia Lower, who was a Masters of Science graduate of the class of 2014. Denise received her undergrad and MS at RUTGERS, RUTGERS University on the Camden campus, where she studied the biodegradation of single-use plastics by white rod fungi. During her undergrad and graduate studies, she taught general biology and microbiology labs and tutored students in ancient Egyptian hieroglyphs. So very broad background and diversified interest. But her current main interests are in biological oceanography, in conservation and wildlife. Denise is currently studying the biology and ecology of sharks and corals while training to become a certified scientific diver. Denise.
Thank you for the introduction. All right. Hello, everyone. As previously stated, my name is Denise Hassinger. I am a Master of Professional Science student at the University of Miami. So a little bit of background information, even though you already got a little snippet of some of it. I completed my undergrad and master's at Rutgers University in Camden, New Jersey. For my undergrad, I stayed very broad with my studies. I dipped a little bit into the ancient Egyptian hieroglyphics. I analyzed starling murmurations. I looked at the converting cassava into a sustainable biofuel, which could be used to help sustain the economies in Western African countries. I did a little bit of anthropology work in Old City, Philadelphia. And I also surveyed the intertidal zones of the Florida Keys over a few summers for my undergrad. For my master's, I focused specifically on trying to see if certain fungi, white rot fungi, were capable of biodegrading plastics, single-use plastics specifically. Now, white rot fungi doesn't necessarily have a pleasing name to it, but we're all familiar with this group because we consume them on a regular basis. So we have pearl oyster, shiitake, reishi, All of these are white rot fungi, and these are the guys that I used to see if they are capable of breaking down these plastics, which I found out they can, which is a really neat thing to utilize for later. Over the years, I spent a lot of time helping out with nonprofits. So one of the new nonprofits that I'd like to highlight is ADFC. So it's a whole bunch of aquarium divers getting together to become citizen scientists so they can start helping out with coral restoration work. And then I spent a lot of time at Freedom Center for Wildlife. So this is a wildlife rehabilitation facility. Spent a lot of years learning how to properly care and handle various native wildlife to New Jersey. And the hawk that you see in the photo here, her name is Cheyenne. She's a very special hawk. She is a Harris's hawk. She's not native to New Jersey, although she resides in New Jersey. But what's so special about her is she is the oldest known captive Harris's hawk. And she'll be turning 36 years old this March. So she is living history, and I've had the honor of taking care of her. So currently in my work, I am in the MPS program. I'm specifically in the marine conservation track, which is where I want to emphasize my career in the marine ecosystems. I am currently studying the biology, ecology, and conservation of sharks and corals. So these are vital animals within the marine ecosystems. And I'm also training to become a certified scientific diver. This year, I was selected to be one of the Cornelia Ware Memorial Scholarship recipients, which I am super honored to have been selected. And it provides vital and critical financial support that allows me to maintain my focus on my studies. And I also received the MPS merit-based tuition waiver, which I'm also thankful for. And then through all these broad studies with my undergrad, my master's, and my second master's here at University of Miami, it leads to my ultimate goal of being able to contribute to the conservation of Earth's biodiversity, specifically by resolving negative human-wildlife interactions. And it'll be through a variation of combination of scientific research and education. So thank you for listening, and enjoy Sea Secrets.
Thank you so much, Denise. Very interesting. Congratulations on the hard work that you are doing, and we are delighted to have you on board in our program. And I am sure that your career will be very, very impressive, as your studies have been. It is now my true pleasure also to introduce Dr. Nikki Traylor-Knowles. Nikki is a cell biologist that studies the evolution of immunity, wound healing, and regeneration of cnidarians, a group of about 9,000 marine species, which includes the corals, the jellyfish, sea anemones, sea reefs, and ferns. Her lab uses technique and cell biology, genomics, microbiology, and molecular biology to study the cnidarian's innate immune system and its interaction with climate change and other human-induced stressors. Nikki is interested in the application of these techniques to conservation efforts through the development of biomarkers for predicting health outcomes in corals in the face of climate change and disease. Nikki has joined the Rosenstiel School in 2016, so that's a little bit over 4 years. She got her undergrad and her master's thesis at Johns Hopkins and her Ph.D. from Boston University in 2012. She did a postdoc after her Ph.D. at Stanford University and back at the Hopkins Marine Station under Stephen Palombi. Even though Nikki has been with us a very short time, she has already demonstrated, I would say, her capability to become a key leader for the University of Miami, and in particular for our school. She has in this short time published more than 33 peer-reviewed publications, which is quite impressive, and received already 3 very important grants that are supporting her research, including one from the National Science Foundation, one from the Florida Department of Environmental Protection, and one from a private foundation named Revive and Restores. Nikki has already obtained a few awards, and it is truly a pleasure to now listen to what she has to tell us this evening. Thanks, Nikki, and all yours.
Okay, thank you. All right. It is really wonderful to be here. I'm so grateful for this invitation and to be able to speak with all of you virtually about the work that I have been doing at Rasmus and beyond, and thinking about not only how my work can help save corals, but also the applications maybe to human health, and what can we learn about human health from corals. If you're interested, I am on Twitter, so please feel free to follow, and I will follow you back. And here's also my email if you're interested in, if you have any questions, please feel free to reach out. So I wanted to give a quick introduction of who I am, because I'm more than my credentials, right? I'm a twin mom and a wife. So my twins are 3 years old and they keep me quite busy, especially right now during the pandemic. I'm a scientist, mentor, and teacher. I take a lot of pride in really mentoring students and helping students succeed in whatever area they're interested in succeeding. And I really enjoy doing science and discovering new things. I'm also an advocate. So recently I've started the group called Black Women in Ecology, Evolution, and Marine Science. And I'll talk about that more later in my talk. But I'm really excited about this because I'm able to use my platform to be a voice and to help bring community to a group of folks that maybe didn't have that community. And recently I've become a special collections director, which has been really challenging, but also really fun. And I'll talk about this more towards the end of my talk about perhaps some of the applications and things that we can discover in this collection that maybe have applications to human health. And this, just very quickly, is my laboratory. These are all the wonderful people in my lab, and we're located on Virginia Key. So when the pandemic's over, maybe we'll be able to visit. And this is our laboratory website. So if you're interested in checking out the work that we're doing, we tend to refresh it quite often. So coral reefs are the foundational marine tropical ecosystem. And I think here in Florida, we know this and understand this because we live by it every day. But maybe if you're tuning in from somewhere like Iowa, this maybe isn't quite as intuitive. So coral reefs provide food, they provide shelter, they provide biodiversity for a bunch of different organisms. And on top of all of that, they're just really beautiful. So if looking at this image, there's just so much going on in here, so much activity, and there's a lot to discover. And so for me, one of the things that's really drawn me to coral reefs are that you're always discovering something new. So you can dive on the same reef 100 times and still see something new each time. And I hope today in my talk, I'll be able to show you that even at a cellular level, we can see and discover new things all the time. So what is a coral? I often get asked this, like, are corals just rocks? Well, they are kind of rocks, but they're rocks with animals on top of them. So corals are colonial animals. And what I mean by that is not that they are from the 1600s, but that they clone themselves. So they start as this one polyp that then clones itself for hundreds and sometimes thousands of polyps that grow together, and they secrete calcium carbonate that creates the structures that you see and you know when you swim on a reef. And they provide habitat for fish and other organisms, just like I discussed before and what you saw in the previous video. And the other thing that's really cool about them are that they have these specialized cell types that are called cnidocytes. And these are one of the defining features of corals and other cnidarians, so cnidocyte, cnidarian. And what's so neat about these cells are that they're like little harpoons. So if you've ever gone snorkeling or diving and you've had the unfortunate experience of brushing up against a fire coral and that feeling that you have of the burning, well, that's a bunch of these cells going into your skin. And so they have different types of toxins and other things that they carry within these cells. And it's a form of protection and also a way in which they feed. So it's a really unique thing that they have. It's very important for the survival of corals. Additionally, as I talked about, corals are these colonial organisms. And so all of these different polyps feed and interact. And so this is a close-up picture you see of one of these polyps. And what's really, really cool is that beneath the surface, these polyps actually communicate and interact with each other. So in this video here towards your right, what we did is we took a piece of coral and we cut it. And what you see, all these kind of wormy looking things, that's actually what are called the mesenterial filaments, the insides of the coral coming out and going into the site where it was wounded to clean out the site. And what's so interesting is that you actually see communication and interaction between these mesenterial filaments between the different polyps. So there's communication that's happening amongst these different polyps and making it sort of this super organism. Additionally, on top of just being really amazing super organism, they're also holobionts. So what does that mean? What it means is that they have multiple organisms that live within them. So on top of having these multiple polyps, they also have a symbiotic algae that's called a dinoflagellate. And the dinoflagellate provides food for the coral and in turn takes the waste and eats the waste of the coral. And so these organisms are really, really important for the coral health. And so many times when you hear about coral bleaching, that is because the relationship between the coral host and this dinoflagellate have broken up. But not only do they have these really interesting relationship with this dinoflagellate, they also have all these other organisms that live within them. So including viruses, fungi, archaea, prokaryotes, all kinds of things that live within their tissues. And so it's a really ecosystem in itself. And it's really exciting because every day we're discovering a new organism that's actually living or coexisting with corals. But the bad news is that corals are under threat, and we're seeing this globally. So this is an example from 2016 of a really horrible bleaching event that occurred on the Great Barrier Reef. But we're also seeing this locally, right? Where right now we're in the middle of not only a pandemic on land, but also a pandemic in the ocean, where we're seeing that this disease called stony coral tissue loss disease has been spreading throughout Florida and the Caribbean, and we don't know the cause, but it is killing many types of corals at really alarming rates. And so one of the questions that I often get is, well, what would happen if corals disappeared? Like, what would happen? So let's look at that. So what would happen if corals disappeared? Scientifically, we would lose a lot of unique animals and also the ability to discover a lot of novel chemicals that could be important for human health. We would also have no more of the beauty, biodiversity, and our coastlines would definitely be compromised because corals can act as a barrier from high wave action. We would also have no more local fisheries. So there would be reduced tourism. And in the end, Florida would really lose billions of dollars a year. And so we're left with this, where we're left with rubble, dead coral, sometimes octocoral and sponges. And while I have nothing against octocoral or sponges, it's just very different from what we know from a typical tropical marine system. So one of the questions that has always driven my work is, why do some corals survive and some don't? So, you know, I've talked about that there is these big stressors that are occurring, causing bleaching and disease. Yet always in these outbreaks, there's either one or two corals that might survive. So the question is why? And so that's where the immune system comes in. The coral immune system is a defense system, much like what we have, but it's a little bit different in corals. So I know right now we're hearing a lot about immune systems because of COVID, right? So it's been in the news a lot. And the idea of the immune system is that it's a defense system against any foreign pathogen or even any type of cell that may be misshaped or misformed. And in humans, we have a pretty complex immune system that has memory to it. So that's where the idea of vaccines come into mind, because you can get the vaccine, it will create immune memory so that you won't be able to get that disease again. But we also have this innate immune system, and that's what corals have, where they have the ability to kill bacteria. They have an epidermis to protect their inside cells, and they also have mucous membranes. So let's look at sort of a simplistic version of this. We have our pretty coral here. And so corals can secrete what is called melanin. And it's actually, melanin is something like what we have in our skin that creates the different shades of our skin. Corals can secrete this as a way to fight against different types of pathogens. They also can secrete fluorescent pigmentation, which can act as a way to kill bacteria and quench different bacteria or viruses that might be affecting the coral. They have this mucus epithelia, and this is really interesting because it's full of a lot of antimicrobial peptides and other things that may be actually really important for human health and have the potential to be developed into novel types of peptides. They also have a lot of different genes and pathways that are involved with immunity that we don't know anything about, but we're just starting to discover. And lastly, they have cells. So they have these building blocks of tissue called cells that go around and migrate throughout the coral and can help to find and attack any type of pathogen. So an example that I like to show is about the immune system. And this is one of the things that drew me to corals, are that they have a really complex immune genes. So if you have no idea what a gene is, or maybe don't care, that's okay. All I want you to take away from this is that genes are DNA, and corals have a lot of different diversity of these important genes. So as a comparison, let's look at humans. So in this, you don't have to know what TNFR is. It's a type of immune gene. They have 29 of those. And so when in my postdoc, when I was studying this, I thought corals will definitely not have more than 29 of these genes. And it turns out that they actually have 43. So they have way more than humans, and actually more than a lot of other animals that they're closely related to. So they have this diversity of immune genes, and we don't know why. So that's one of the things that I'm studying and my lab is studying, is trying to figure out why all this diversity, and what is it, is this one of the reasons that they've been able to survive for so long? And how is this affected by climate change and disease? So corals are being heavily impacted by disease, and this is so apparent here in Florida. Over 50% of the world's coral reefs have died due to climate change and disease. So it's pretty dire. And we're in a race against time, really, to save corals. And so these pictures that I have here of corals that are experiencing different types of diseases. So this one here is a coral in Florida that is bleached. This is another coral that is experiencing stony coral tissue loss disease. And you can see that this is the skeleton underneath, the tissue's just being rotted away. And this is another disease here called white band disease that is again rotting away the tissue of this coral. We're seeing that there's an increase in disease virulence and abundance due to human-induced climate change. And this is really challenging if you're trying to save a reef, which many of us here in Florida are working actively to restore reefs. And so there's a real push to understand these diseases and understand ways in which to mitigate these diseases. But coral diseases are really frustrating to work on. They are, a lot of times we don't know what is causing the infection. So the infection that we might see in the field, is that due to is that the primary cause or secondary cause? Is the disease itself, what is causing it? Many times we have no idea what is causing it. Also, IDing the different diseases can be very challenging in the field. So we're, in scuba diving or snorkeling and trying to identify these, and many times the same disease can look different on the same coral species. And lastly, being able to actually what we call culture or grow the pathogen in the laboratory to study it more closely is extremely challenging also. And we haven't had a lot of success with that because it is so challenging. And so we have a hard time right now trying to figure out what these diseases are. And so one of the things that we're now trying to do is use clinical aspects to study coral reefs and study disease. And this is just an example of some of the frustrations. So this is white band disease. And my student Ben did this, and I love this slide because I think it really encapsulates what we're up against. So for this one disease, there's over 11 different types of bacteria that have been identified as possible causative agents. So it's really tough to work in this environment. And so what we're working on in my laboratory and in other laboratories as well are to develop these clinical tools for assessing coral health. And so what are these tools? Well, they include something called histology, which is the ability to look at the tissue. So this is a preserved tissue, and you can take a section, a very thin section, and look at the interaction of the different cells. We use enzymatic assays. So this is another way in which to test and look at the health of the coral. So the enzyme can be a particular marker for immunity, and we can look at how corals react in the face of disease with these different enzymes. Recently, within the last 10 years or so, 15 years, omics, or looking at the DNA of corals, has become very, very powerful and popular. And so this is powerful because you can get a huge view of what is happening with all the different thousands of genes in the coral. And also recently, live cell imaging and isolation have become very important for looking and diagnosing the coral health. And so my laboratory is really using both this omics approach, so looking at the DNA, as well as flow cytometry to look at live cells and develop live cell assays so that we can better diagnose the corals and not have to go out and collect them and possibly harm the corals that are in the wild right now. So in my opinion, this is really the frontiers of coral health diagnoses. So developing these live cell assays to assess the coral health, understanding the mysteries of the genes that are involved with these different disease states that we're seeing, and then creating some sort of diagnostic in which that can quickly be used and effectively and efficiently used, particularly in Florida, but beyond Florida. So I quickly just want to run through kind of a case study here, where when I came to Florida and heard about all the different diseases that were happening, my first thought was, well, maybe these corals are immunocompromised. So maybe their immune systems are actually depressed, and they're not able to respond to changes in their environment. And so that's why they're getting all these diseases. And so to examine this, we're going to look at two different species. This is called Acropora palmata, and this is Acropora cervicornis. Both of these are important keystone species in Caribbean systems, and particularly here in Florida. And so if you've ever gone diving in the Keys, you may have seen some of these. And I think the next Sea Secrets, I believe, or the one after, they're going to, with Diego Lehrman and Andrew Baker, they're probably going to talk about some of the restoration efforts that they're doing with these different corals. So what we did to assess this question of, are these corals immunocompromised? We did a disease challenge experiment. And this was in collaboration with Margaret Miller, Dana Williams, Stephanie Rosales, and Zaimara Serrano. In this experiment, what we did is take a piece of healthy coral and graft it to a diseased piece of coral. And the diseased piece of coral, you can see here, if you see my, my arrow, this white part is the dead part. This is the part through here, the disease front. And after 7 days, we look at the coral, and what we see is that the disease has been passed to the healthy piece of coral, and it's actually gone all the way up the coral to transmitted to the coral and is slowly killing the tissue. And so then what we did is we took this and sampled it and sequenced it for its DNA. And we wanted to look at the immune genes. And bam, I know you're looking at this and you're saying, oh my gosh, what is on there? This is like, there's a lot. I put this on here purposely to have that effect. What I want to show is that they're mounting an immune response. So what was so surprising and cool was that we saw that all of these are all genes, and this represents if they're on or off. So red means it's very much on, blue means it's very much off. And we saw that there are actually a lot of genes that are reacting to the disease. So that was exciting, and this is part of the work by Ben Young and his thesis project. And so the next part of this was, well, let's look at the live cells, right? And so this is where we use this technique called flow cytometry. So let me explain what this is. So I'll go back. You have a cell, and with corals, they actually express and have natural fluorescence in their cells. It's really cool. So if you look at them under a UV light, you will see that they just fluoresce. And that's like what's in my background right now. That's just natural fluorescence. So we can use that to separate out different cell types. We can expose them to a stressor, and we can put them into this system called a flow cytometer that shoots these laser beams at the cells. And as it shoots the laser beams, it will separate out the cells can be separated out based on their size, their shape, and any other types of properties that we want to separate them for, if we use a dye, for example. And so we did this, and we did this with corals that had a disease. And what we saw, we did an assay for immune function. For the immunology people on here, we did a phagocytosis assay. For folks that are kind of new to immunology, just understand it's an assay to look at immune function, right? And what we saw was that there was actually more activity in the diseased tissue than in the healthy tissue or the controls. So this was really exciting because, again, this was pointing to the idea that these cells are not, the corals are not immunocompromised. The immune system is working. So that gives me some hope. And this work, I should say, is done by Grace Snyder, who's another graduate student in my laboratory. And so this matters to me because this is good news. The corals have an immune response, and something's happening, something's going on. So there is some hope. Now, the question is, how are we going to deal with this in terms of the environmental stress that they're dealing with that they can't control? They're trying their hardest to fight all this. Now we need to step in and start doing what we can to preserve them. So I really think that this is the way in which to start looking at the cell expression and linking it to the genes. And then we can start characterizing and understanding different cell populations, including the immune cells like we see here in this video. So taking a step back now, I've kind of talked about, what we're doing to help save corals. But another angle that we can also look at are kind of novel insights that we can gain from the coral immune system for human health. And so I just want to show a few little vignettes. This is a relative of corals. It's a sea anemone called Nematostella. And what's really cool about this is that if we cut this anemone in half, what will happen is, if you look in this video, it will actually regenerate its tail, or physa, and regenerate a head. So in this case here, this one is actively regenerating a head. And you can see all this movement that's occurring. It's all the cells interacting and reshaping themselves in order and reprogramming themselves to create tentacles and to create its head. And so you can see it's already created those tentacles and it's moving around. And this one has already healed its tail. And so this is really interesting. And so one of the things that we're interested in is, what are the genes that control this? Are there ways in which to manipulate this? And how could this apply to human health? While I don't know that we'll ever get to the point where we can actually regenerate our own heads, but maybe there's something that this could tell us in terms of wound healing response and regenerating, for example, things in our skin. Additionally, there's a lot of potential for novel discovery of antimicrobial peptides. So antimicrobial peptides act as either a neutralization, or they change the immune system so that it can better deal with specific types of microbes. And so in 2 of these corals that we've been working on in my laboratory, we've been able to discover some novel peptides that could have a lot of potential for being important for different types of bacterial responses and other things in humans. And so that's kind of a frontiers that we're hoping to develop in the future in my lab. Additionally, and recently what I alluded to in the beginning, I've become the collections director of the Voss Marine Invertebrate Collection. So we have this amazing collection at Rasmus that was started in the 1950s by Gil Voss, and then was taken over by Nancy Voss, who ran it from 1989 till she passed away in 2019. And I was privileged enough to be able to work with her for the years since starting at Rasmus, and that she entrusted me with this collection once she retired. And so this collection has thousands and thousands of animals in it. And we're working right now to preserve and digitize this. But I do think that there's the potential for novel discovery within this collection. Not all of the specimen have been preserved in formaldehyde, so there is still some potential to potentially get some DNA. And then also on top of that, there's such a biodiversity, there's so much we will discover in here. So a lot of these things have not been taxonomically characterized. So we're really excited about this and hope that we, myself and my team, Dr. Maria Corrales, who is the collection manager and crustacean curator, as well as Angela Clark, who is the Rasmus library director. The 3 of us are working together to preserve and digitize this collection and be able to share it more widely. So be on the lookout for that. And lastly, I just want to talk about a group that I recently founded called Black Women in Ecology, Evolution, and Marine Science. So I founded this because I was really tired of feeling alone and hearing that we don't exist, but it turns out we do. So there are over, we started in the summer, there's over 200 members, and the members are from undergrad level all the way up through professor and beyond. And we have monthly virtual events and socials. And right now I'm in the process of planning along with the planning committee of having a year-long event of virtual science shares, virtual workshops, professional development workshops. So if you or someone you know identify as a Black woman in ecology, evolution, or marine science, please connect. So we're on Twitter. You can also email us, or you can use your phone right now to scan this and it will send you to a sheet where you can sign up. Alternatively, you can also just contact me and I'll make sure to get you on there. And so with that, I thank you for being here and for going on this journey with me about corals and how amazing their immune systems are. There's a lot of people that I want to thank, including all of the lab members, Ben Young and Grace Snyder, of the work that I featured today, as well as I'd really like to thank Dean Ronnie Abisar, Deanna Udall, Jennifer Dillon, Josh Coco, and Catherine Gracioso for their help with this whole presentation. And lastly, I'd like to recognize Maria Crialis and Angela Clark for their help with the marine invertebrate collection and all of the collaborators that helped make this work possible. And so with that, and my funding, I should say too, thank you. And so with that, I'll take any questions.
Wow, Nikki, fantastic. I knew that you were good, but you are very, very impressive. That was a wonderful, wonderful presentation. Thank you so much, okay? And certainly, you know, from where I sit, it's a true pleasure to see how in so little time you have evolved to become a giant already in our school. So thank you for being here and for agreeing to do this presentation. It was fantastic. I want to, before I open that for questions that are going to be led by Josh, Diana, and Kat, I want to indicate that we have the next lecture is going to be on February 9, and we are going to hear about our Cooperative Institute for Marine Atmospheric Science Studies, CIMAS, which is our collaborating center with the NOAA. And that will be on February 9. And again, Nikki, you forgot to mention the cat, okay, with all the responsibilities that you have. But again, thank you so much. And it's open for questions.
All right. Good evening. Thank you, Nikki. That was really great and very informative. So we've got a couple questions, actually some really good questions.
Yeah.
We had over 450 people that logged on and attended. You broke new records, so thank you. Thank you to all that have attended. So first question comes from Macarena Blanco. Do the mesenterial filaments have also competition or attacking functions? She says she saw them deployed when two colonial fragments were growing together and wondered what would be the reason.
Yes, so they do have, when you put two different corals next to each other, they will extrude their mesenterial filaments and fight each other. So there's a lot of defense mechanisms that are involved with it, but also I think in some species, and they're also involved with digestion. I mean, they're kind of, they do a lot of things. So yes, what you saw was a defense mechanism.
Next question we have is from Joyce Nelson. She says she's been fishing on the flats for over 20 years in Biscayne Bay. Now there are no fish and seagrasses are gone. She believes it's due to pollution and plastics. In the past, when we went to the flats, we would also see sharks, rays, bonefish, starfish, permit, and snapper. Now there is nothing. Even the lobster are gone. How can we recover from this horrible times?
You know, that's the $1 million question, right? This is a human problem, right? And so the actions that we do, and then actions at a more broader level, government level, are really important. And so, you know, I get asked this a lot, like, what can I do personally? Because I think climate change and other issues they're such big issues that it's sort of hard to feel like you can do anything. And I think that I personally believe that your actions matter. And so, you know, making informed choices about the food that you eat, for example, or not using disposable items is another thing. Thinking about what you're pouring down the drain, especially here in Miami. All these things, while they are little and they're not going to completely solve the problem, but they can help. So, one of the issues in the Bay is that we're dealing with overall climate change, but we're also dealing with a lot of local issues. And so the other thing that I would suggest is contacting, your local politician or commissioner and making sure to have a loud voice and be known that you care about these things, because I think that's sometimes neglected, that we do care about this. And so, politicians need to hear that.
Excellent. Another question from Christine Moog. Sea star wasting disease along the West Coast was found to be related to the effects of climate change, which seems to suppress these animals' immune system. Is the similar thing happening in stony corals?
So this is the interesting thing. When we look at the genes of corals that have been exposed to stony coral tissue loss, they are having an immune response, but not in a typical way. So we're still trying to understand what is going on. It looks like there is some complex mechanism that's happening that involves possibly the symbiont that lives within the tissue, the dinoflagellate. And so, yeah, so the jury's still out on what is actually causing it, but we're actively investigating to figure out what the immune system is doing.
Here's another question from Diane Marie Brock-Smith. She asks, have there been attempts using coral progenitor cell lines and exposing them to possible pathogens?
Progenitor cell lines. So we don't have any progenitor cell lines. I wish we did in corals. That's one of the things that we're working on. So we're working on developing and identifying and characterizing stem cells in corals. So we don't have any like immortal cell lines or things like that we can work on within corals. So that's one of the hopes in my laboratories, to be able to start develop these techniques to do more targeted approaches like what you do in medical science, right? Instead of having to take a whole piece of coral and work on it, we can just work on the cells and get answers much more quickly. So that's where this is going.
Are any of the coral disease autoimmune diseases, as in component of the holobiont has
Yep.
ceased to be in benevolent? In the same line, do coral cell become transformed?
Okay, so right now we're still trying to understand that interaction between a lot of these diseases and whether there is some sort of immune relationship with why it's taking over. And we don't know, but what we are seeing is that they are not compromised. Some of these diseases may be related to the holobiont. And that's something that we're also actively looking at because the holobiont, that algae that I showed that image of, there's a lot of bacteria that live in that cell and around there. And so maybe those are a potential source for different types of diseases. So we're exploring all of that. I can't give a definitive yes or no answer, but we're actively looking at that.
Rachel Rode asks, do you think grafting species with a stronger immune response to diseased corals might help weaker corals survive?
That's interesting. So I think that the problem that we have is that a lot of corals also have a what's called allorecognition response, where if you put individual A and individual B next to each other, they will have an immune response because the tissue is seeing that it's right next to something that is not itself. And so I think that could be a problem. But what could be interesting is if we could, and something that we're actively working on, are using stem cells as perhaps a therapeutic for inducing more resilient corals. So we're in the beginning stages of that, but I think it's one of those kind of cool sci-fi, but also really applicable things for helping to save coral reefs.
Excellent. And finally, we have a question. Would undergraduate students be allowed to get involved in the research?
Yeah. So I have, currently I have 4 undergrads that are doing research in my laboratory. Right now, because of COVID, I'm not accepting new students, but in the summer, I'm hoping that I'll be able to, because then we can hopefully by then, you know, be able to have people in the lab and with all the safety protocols and things of that nature.
We have a question from Lisa Gethard. It says, regarding your studies at the coral reefs and immune system, are there antibodies such as those found in the manta ray in southeastern region of Florida that could be used in research for the COVID-19 and mutations?
Possibly, but we, I mean, corals themselves don't produce antibodies in the same way. So I think more of like antimicrobial peptides might be something that we could discover, other novel peptides. Corals don't have the adaptive immune system like vertebrates do that can produce antibodies in that way. But I think more of what could be discovered are these different types of chemicals or things that maybe could be used in antibiotics, for example, and other things of that nature.
We're getting many compliments about your cnidarian shirt, your blouse.
Thank you. Yes. I wore this especially for this presentation.
And another great question from Tomoko Bell. It says, I'm wondering why corals were able to develop more immune-related genes than humans.
I wonder this too. That's why one of the reasons I'm working on them. So, I mean, I have a few theories about it. I think one, they're really old. So from an evolutionary time point or from an evolutionary perspective, they've had a long time to develop a system, right? So I think that's one aspect. I think the other thing is that they live in company with all of these important and different microbes. So they have to have a system that can sense these different things that they're living with. So those are my hypotheses, but again, this is what we're studying is why, because it's really surprising, but also really cool.
Awesome. Rodrigo Zuniga wants to know, what is the difference between a primary and a secondary coral infection?
Right. So a primary, what I meant by that is that, so a primary infection would be something like you get in the case of, let's see, you can think of it like this. It's almost like with AIDS. Many times people die not from AIDS, but from a secondary infection, right? From some other infection that they get because their immune system is so shot due to AIDS. And so it's sort of similar with corals, that it may be that there is a infection that they're getting secondarily. So they get a first infection, some sort of bacteria maybe, and then that causes their immune system to be a little bit reduced, and then let in more, maybe a virus could be carried in. So that's what I mean by primary or secondary. And so we don't know with a lot of these diseases what is the cause.
Mm-hmm. So we have a question from Mike Jasper, who I believe is a parent from one of our current undergrads. Is there a geographic component of where and how these diseases get established, or are they universally present around the world? Great question.
Yeah, so there is geographicness to them, and there's also specificness to different species. So for example, the stony coral tissue loss disease that we're seeing right now, it affects over 25 species, but they're only species that are the bouldering type of coral. It doesn't seem to affect like a croprid branching species, and we don't know why. So there's definitely a geographic element, but there's also like a species or physical element to many of these diseases.
And I'm going to give you one last question, and then we'll wrap it up. But Mariana Leota asks, can the holobionts be compared to human microbiome? So if there is a imbalance in the microbiome composition, the risk of an overgrowth of pathogens increases.
Yes, exactly. That's exactly how it is. It's sort of, there's a, what we call homeostasis, right? There's a balance. And so in the case of corals, when we have heat stress, when we have pollution, whatever, what we see is that there will be a imbalance that can occur, which can cause bad bacteria or whatever, more pathogenic bacteria to grow on the coral. And so the question is, though, why does that happen? You know, what is the mechanism of that? And what does the immune system do to either facilitate that or help or stop it? So that's one of the areas that we're actively researching.
Excellent. And last question from Lisa Gethard. I am amazed by the visual you shared with the regeneration of the head and limbs of the coral. Would you consider a study such as this in microgravity to research the cell replication process?
Sure. Please reach out. I always love working, as you saw from my thank you slide. I have a lot of collaborators, and I'm always excited to try novel and different things. And I think because corals are so impacted, we have to be creative and use different strategies. So yes, please reach out and let's try this.
Excellent. Nikki, thank you so very much. We are very grateful for the time and all of your incredible research that you're doing. We will see everybody on February 9th for CMAS, which join us will be our NOAA Cooperative Institute for Marine and Atmospheric Studies. Anybody who registered, you'll be getting an email with a link to the video, but also we will share whatever questions we didn't answer with you as well. And please reach out to Nikki. She shared her information. So if you have a question and you'd like to follow up directly, please do so. All right, I guess with that, we're good. Thank you all for attending. Thank you so much.